Hello, everyone. Good evening, and welcome to this third appointment of the cycle of seminar by the
This
Italian
conference
Geological
will now be
Society.
recorded.
The overarching theme of this series of webinar is geobiology, and it is my great pleasure today to introduce Paul Falkowski. I've known Paul for a number of years now. I was just a visiting PhD student at Rutgers when I met him the first time, and he's been one of my mentors and has greatly influenced the way I approach science and my line of research right now. Paul graduated from the City College of New York, and then he moved on into getting a PhD at the University of British Columbia. He then did a brief postdoc before being hired by the Brookhaven National Lab, and then later on he moved to Rutgers University, where I met him and where he is today. He has an impressive number of contributions to very different fields, from photosynthesis to biophysics to the way coral precipitate calcium carbonate to biogeochemistry and the nitrogen cycle. So it's been very prolific in his career. He produced a lot of good ideas and very influential papers. And today, Paul is going to speak about the coevolution of geosphere and biosphere, thinking a lot more about the evolution of oxidoreductases and redox reaction in life, which is a topic that I find very fascinating, and I've been learning a lot from Paul. And there's a lot more I could be saying about Paul, but we can leave that for conversation another time. And thank you very much, Paul, for accepting our invitation, and I'll leave you the floor for this presentation.
Thank you, Donato, for that very kind introduction, and I thank the organizing committee for inviting me. So I've been interested in the evolution of life since I'm a child, obviously, but we avoid those topics broadly until you get to be somewhat mature in science and you have a long record. Otherwise, you're not going to be funded, at least in the United States. So why is my screen not working? Here we go. So most of what I'm going to be talking about is from a program that I lead called The Evolution of Nanomachines in Geospheres and Microbial Ancestors. And if you're interested in the team and what each of the individuals does, there is the link. We shortened this to a very simple acronym called ENIGMA. And if you think about it, we know where we got to. So if you're familiar with New York City, this is obviously one half of the George Washington Bridge, a very famous bridge in the United States, which goes from New York City, from where I'm shooting, to New Jersey, where I live. And it crosses the Hudson River. Now, if I didn't tell you that, you wouldn't know what was on the other side of the bridge. So you know what's on the side of the bridge where you can see now. So that's life. We know that. We got there somehow. How we got there from the first side of the bridge, which we can't see, we don't know. So how this bridge was constructed remains one of the great, what my friend Nick Lane would call, black holes of science. It is one of the most profound questions which has been out there for more than 100 years, since Operon, actually just about, Operon wrote his first paper on this in 1924, I believe. And Fox and others have been working on this for many years. But the idea of prebiotic chemistry we've had for a very, very long time. Going from prebiotic chemistry to something that's actually self-replicating, autocatalytic, is one of the great enigmas, and to borrow the pun of the name of the program, of life. Now, I'm going to be talking a lot about specific metals, and one of those metals is obviously going to be iron. And here I am at the top of the Ishua Formation in Greenland. This is in southwest Greenland. There are no roads there, so you have to go there by helicopter. But what you can see very quickly if you're a geologist is you can see these laminated rocks, which obviously were sedimentary. They've been metamorphosed to some extent. And there were marine sediments. And what you can also see here is there is a lot of iron in these sediments, a lot of iron. So these rocks are 3.8 billion years old, and the iron precipitated. This is not truly banded iron formations like you would see in Australia, but what you can see is sedimentary material that has more organic or a little bit less organic material in it, and the amount of organic material in this portion of the blacker region is very, 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 very small, and the only real organic, quote, organic carbon in there is graphite. So there are no fossils in here, but I want to show you that iron was very, very, very abundant in early Earth's oceans. So if we start, and I don't know how many of you geologists are familiar with this period of Earth's history, we start the Archean arbitrarily at 4 billion years ago. It ends at an unconformity at 2.5. So it's not truly dated at 2.5. In some places it's 2.52. In some places it's 2.49. But it is a true geological unconformity that leads to the end of the Archean and into the Proterozoic. Now, during this period of Earth's history, there was no oxygen. Now, I won't say no as in 0, 0, but the concentration of oxygen was on the order of 0.0000, 4 zeros, 1%. Okay, this is, I'm not going to make this relative to the present atmospheric values levels right now. It doesn't matter when you're talking about such incredibly small concentrations of oxygen, whether you pose it to the present atmospheric values or the absolute value, it doesn't matter. So with that, the absence of oxygen, UV light could come to the surface of the planet. And this is the solar radiation at the top of the atmosphere. Obviously, there are many, many lines in here. These are, well, there are many lines. There's the line, the Fraunhofer lines, the Lyman lines. This is how we identified elements that we couldn't find on Earth originally. So helium is an example of an element which was identified from solar spectra before it was discovered on Earth. That's why it's called helium, right? For Helios. Now, at the end of the day, the UV light, which is here, could penetrate to Earth's surface. It doesn't do that anymore. It's blocked by ozone in the stratosphere. You don't get ozone until you get oxygen. So I'm not going to go into this whole rigmarole, but the point is that we know we went from a mass independent to a mass dependent isotopic fractionation of sulfur in pyrites at about 2.4 or 2.35 billion years ago. And that's called the so-called great oxidation event. But up until that point, UV light could penetrate to the surface of the planet. And just to let you know, I mean, we can talk about faint young suns and things like that if people have questions, but these lines here are gas composition lines out in the IR. So mostly water, carbon dioxide, a little bit of methane that you could see, but it's very difficult to pick it out from here. And UV light actually penetrates in the ocean quite deeply. It's sometimes a misnomer that people think that UV light doesn't penetrate very much in the ocean. It penetrates pretty easily when there's no organic matter in the ocean, when it's just water. So why do I care about UV light? Because every single covalent bond in your body, in my body, in every animal's body on this planet, every single covalent bond is a relic of solar energy conversion through photosynthesis. All life on this planet, all life is driven by solar energy. And if we want to talk about chemosynthesis, we can talk about it. But at the end, the redox gradients for chemosynthetic organisms are virtually all of them are based on solar energy conversion somewhere else that made a redox gradient, even in deep sea hydrothermal vents. Now, this is a very, very old paper. It's from maybe 2016, 2017, from one of my former postdocs, John Kim. And what we did here is we made siderite, which is iron carbonate, a very abundant mineral at the time in the Archean. So siderite is not stable in the presence of molecular oxygen. It will decompose because the iron will become oxidized from Fe2 to Fe3. But let's take a look. Here it is under an anaerobic condition in aqueous phase. And we're going to blast UV light right onto that little part here. Now, we don't do this except for this demonstration. We don't do it like this. But I want to show you that light can oxidize the iron. And the oxidation is occurring at about 270 nanometers of light in the UV, but not very deep in the UV. So this light would have been very available during the Archean. Why am I going there? Because we don't often think of light as a driver of mineral change in Earth's history. This was discovered many, many, many years ago by Karen Smith and others. And Karen Smith wrote several very profound papers about this, were published in Nature, and they wrote a book about it. But Karen Smith died, and a few other people have taken it up, Martin Schunan, and now in our lab, we do this a lot. So we have a much more efficient system of irradiating minerals in general with UV light and looking at the transformations. All right, let's continue on with this. So this is the transformation. We made the oxidation of siderite into a new mineral, which is magnetic. And you can see the X-ray diffraction over here. This is just a bar magnet. So we can make hematite and magnetite just with light, no bugs. And from this, we get a production of hydrogen gas. Now, why do I care about that? Because this is a mechanism by which hydrogen gas is supplied to the surface of the planet, independent of volcanoes. It actually, if you do the maths, which we did in the paper which was published on this photo-oxidation of siderite, when you just lay out the siderite in shallow seas across the world at the time, you get a production of hydrogen gas that is very, very similar to that of volcanism, which is mind-boggling. So why am I doing that? Because I want you to think about the earliest metabolism of life. The earliest metabolism of life almost certainly was based on the oxidation of hydrogen gas to generate protons and electrons, or using the hydrogen directly to fix carbon dioxide to make organic matter. Now let's go on. So this is the classic Goldschmidt type plot. And here's hydrogen helium, obviously. These are primordial. These are going back to the Big Bang. And then we come to the lithium beryllium boron hole, which I'm sure you've all in geochemistry explored why that is. Why do we have this lithium beryllium boron hole? And then we come to the carbon, nitrogen, oxygen, up, down, up, down, up, down, the odd, even, odd, even numbers of protons, which is a pattern that is throughout this entire graph. And then so let's just take a look at the big 6 elements of life. There's hydrogen, most abundant, carbon, nitrogen, oxygen, second most abundant, carbon is the third, nitrogen is the fourth, then we come to sulfur, 5th, and phosphorus, the least. All right. All of these are made, obviously, in supernovae. They're not made in our star. They're made in very, very hot, very short-lived stars. And some of those stars are cooler or shorter lived than others, and they explode, and the element that they leave at the end of that explosion, to the most part, is iron. And then we get into other reactions, and we can get onto neutron stars, which are giving us radioactive elements, uranium, thorium. So just remember now, we have 2 sources of energy. We have fusion energy from our stars, which is hydrogen to helium fusion reactions. And we have fission reactions, which is in the core of our planet. For example, uranium, thorium, and potassium-40 are giving us heat, which is allowing us to have tectonics. And without that, we wouldn't be here. And I'm going to ask the graduate students in the audience here to tell me, there are two elements that seem to be missing. I think it's atomic number 43 and atomic number 62 or something. Is it because these elements are not real? Or what is the reason that they're not there? So just think about that. So the invention of the origin of life requires a redox reactions, and I'm going to explain this very quickly in a sentence or two, so you remember what redox reactions are. Most geologists learn oxidation reduction reaction, learn acid-base reactions, and they forget a little bit about redox reactions. So there are redox reactions between hydrogen, carbon, nitrogen, oxygen, and sulfur. Phosphorus not. Phosphorus undergoes acid-base reactions. It can undergo, under extreme conditions, oxidation reduction reactions, but for the most part, it participates only in acid-base reactions in life. So what is an oxidation reaction? An oxidation reaction is the extraction of an electron with or without a proton, or the addition of molecular oxygen to a molecule or atom. If it is just the addition or subtraction of protons, that is an acid-base reaction, unless it is accompanied by the addition or subtraction of an electron. So we're looking at electricity. This is not a metaphor. Oxidation reduction reactions far from equilibrium are generating electrical fields. So life is electric. So all organisms derive energy from for growth and maintenance by moving electrons from substrate to product. And substrates and products then ultimately must be cycled. Otherwise, you run out of them. So biological processes, just like oxidation reduction reactions, are paired. They're half cells. So you're a half cell, I'm a half cell. We're breathing in molecular oxygen from the room. It wasn't generated in the room, but it's in the room. We're breathing out two gases. We've reduced the molecular oxygen to water, and we've oxidized in our bodies carbon from the sugar we ate yesterday and produced carbon dioxide. So there we become, in a global sense, an oxidizing agent of carbon. We're a reducing agent with respect to oxygen. Now, photosynthesis and respiration are two processes which we all know are paired, and we learned this somewhere maybe in the 4th grade. And let's just take a look at what the electron source today is. So the electron source today is liquid water. It wasn't always so. But let's take a look at the liquid water on the planet. So this is a not photoshopped image from Apollo 11, I guess it is, looking at Earth. It's a beautiful image. They actually used film in those days. They used Hasselblad cameras with very wide lenses and very large films to get the best resolution. And if you are ever in the United States, in Washington, D.C., the Space and Science Museum, you can actually see many of these images. Very beautiful. So you see all this water, and we all know that 71% or whatever it is of Earth's surface is covered with water. Big deal. If I took all the water and I rolled it up into one ball, that's the volume of the water that you would see. So I'm going to ask or just tell the graduate students this. If the diameter of the Earth was represented by 1 meter stick, the thickness of the ocean would be equivalent to approximately 2 typewriter pages. It's not even close to a millimeter. That's how thin the film is on the planet. And I'll just tell you now that that water was delivered almost certainly, mostly by meteorites, not by comets, although some of it maybe have been by comets too. But it's mostly, the DH ratio is mostly corresponding to meteorites. Now, as per Vernadsky, all living organisms exchange a gas with their environment via redox reactions. So you all know who Vladimir Vernadsky was, I guess. If you don't, he was a Ukrainian who lived in Moscow. He was Russian mostly. And he was the father of, in effect, geochemistry. He wrote a book which was published in Russian, and it wasn't translated until, he wrote a book, I think it was published in Russian in 1926. It wasn't translated into English until 1998, I guess. So many of the core metabolic processes of life are related to gas exchange. And that's how we can, to the first order, try to understand whether there is life on a planet that we'll never get to, an exoplanet, because of changes in gases that are far from equilibrium. That's the basic idea. So let's just take a look at this hokey little figure here, which I'm sure you all know in geology. So this is water and carbon dioxide. That's photosynthesis. And we're going to make a reduced carbon, that's sugar. Just make it to be sugar. Chocolate bar, okay? Here's a chocolate bar. And we generate oxygen waste product. That's what oxygenic photosynthetic organisms do. So the leaves on the tree outside in Naples or Rome or wherever you are, that the grasses that are still maybe alive till the winter comes, they're doing that. And then you're doing this. You're taking the sugar, you're oxidizing it with oxygen, you're producing water and carbon dioxide, as I just said. Now, I just want to make a very simple point, which I'm sure you learn in geochemistry. That is, you would have no molecular oxygen on this planet, or very, very, very little, unless you hide the candy from the children. Where do we hide the candy? In the lithosphere. We hide it in rocks, and it has to be uplifted out of the Wilson cycle from the oceans onto continents, because over long periods of time, all we would do is subduct organic matter in marine sediments. It would be oxidized in volcanoes, and it would just we would just generate very little oxygen. So plate tectonics plays a very key role in generating or allowing the planet to generate oxygen. It's not just having this reaction, which is a necessary but not sufficient condition for generating molecular oxygen on Earth. So now there are a selection of these nanomachines that do the work. These are the guys that are actually moving the electrons around, and they're done by catalysis. So catalysis is a system where you lower the free energy of a reaction, and the reaction then can proceed at a more equivalent thermodynamic rate. So when I say thermodynamic rate, it's a little bit of a contradiction because kinetics and thermodynamics don't necessarily correspond. But the point is that it allows a reaction to proceed by lowering the energy barrier. So there's always an energy barrier from a substrate to a product. And that energy barrier is lowered by catalysis. In the true term, the catalysis, the catalytic system is not itself consumed. So here's an example, a little hokey one, of the machinery that generates the oxygen. So the machinery that generates the oxygen is the most complex energy transfer machinery on the planet. It contains two reaction centers. That means that two systems that independently use light. So this is light. The light is, I'm not going to go through the whole thing, but ultimately it oxidizes manganese atoms, and I'll show you how that works in a second, in a special cubane structure, which is contained within these little proteins down here. And it does this one electron at a time. So 4 photons, 4 electrons. That's an Einstein rule. One electron per photon. You cannot generate 2 electrons per photon. You can only generate 1 electron per photon. All right, then we come over and we're going to generate some more electrical energy with more light, and we're going to drive the electrons and the protons back together to a hydrogen carrier, which is NADPH. I'm not going to go through what this means. It doesn't matter. But there's no free hydrogen that is ever made in the system. If free hydrogen were made by this, because you want the hydrogen, that's what we're splitting the water for, to get the hydrogen so you can reduce the carbon to a sugar, for example. If you made the free hydrogen, the hydrogen would escape out of the cell and it'd be lost because its diffusion couldn't be controlled. So it's always the electron is decoupled from the proton and then recoupled to the proton with a hydrogen carrier. All right, I'm not going to go through the rest of this machinery, but this machinery actually works it physically moves. So when this so-called D1, D2 reaction center proteins here, when they're doing an oxidation reaction of manganese, generating an oxidized manganese atom, which is going to further oxidize the water ultimately, they actually collapse. And if you do this and synchronize these with flashes, and you put a little hydrophone there, you can listen to the sound wave. So these guys actually clap, and you can determine the efficiency by the sound. It's what's called the photoacoustic effect. It was discovered by Alexander Graham Bell, obviously not because of photosynthesis, but the photoacoustic effect was one of the major ways which he thought he could build the first telephone. Now this is what we're at now. So these are now the structures of that complex at atomic resolution. And how are these structures obtained? Well, originally they were obtained by an extraordinary laborious crystallization, which is very difficult for membrane-bound proteins. And so this little green band here, those are membranes. So this is the outside of the membrane, this is the inside of the membrane. Very, very, very laborious. Today, we have cryo-electron tomography, which can get you down to about 2.3 angstroms resolution, which is virtually atomic. And you can do thousands and thousands and thousands of analyses of particles on membranes. You don't have to crystallize them. And then you can obtain these structures much, much more efficiently. Now, I just want to show you, this is the blow up right there of the first mineral that you're going to see in this paper. I mean, in this talk. So there are 4 manganese atoms, 1, 2, 3, 4, and 1 calcium atom. This is the rock part that is making the splitting the water and making the oxygen. That's it. That's where it's happening. I'm not going to go through the chemistry, obviously, of this. Is not that critical to you. I will just tell you this. No one knows where this mineral came from. This is not a mineral that is very common. In fact, it's not found that I can find. There's a mineral called, I think it's Moroccanite, which has a very similar structure. It does not split water by itself. I've tried. So if you want to change the world and you're a geochemist, and you can make a catalyst with these kinds of materials, you can make hydrogen gas. You don't have to have that hydrogen carrier. You can change the world by just using water as a source of hydrogen gas, and you can just generate enough hydrogen gas to power the world without anything else. You don't need fossil fuels. And I'm going to give you a little sidebar here. There's enough bond energy in a swimming pool full of water, an Olympic-sized swimming pool full of water, to power the city of New York for a day. So that's an amazing amount of energy. All right, now this is an interesting slide. I think, Donato, you may have been on this paper. So we go from what we think was probably the origin of life-ish, around 3.8 billion. We have no evidence that there's really life then. We don't really see fossil evidence until we get up to about 3.4, 3.3. But in any event, we have reduced substrates that become more and more and more oxidized. Following the great oxidation event is one area. And then, of course, in the in the Precambrian period around 650 million years ago, we get a second burst of oxygen with the evolution of oxygenic photosynthetic algae, not cyanobacteria only, but eukaryotic algae. And then, of course, we get a little bump up with the evolution of trees. And here are metabolisms that follow the change in the redox potential between the substrate and the product. So the lower the redox potential, the more reducing it is. And then the higher the redox potential, and you can see photosystem 2, that's the one right there. That's it. Has an incredibly high redox potential on this scale. It's up to 1200 millivolts. This is the highest redox potential of any catalyst on the planet. Now, what I wanted to do was to do what's called a flux balance analysis of the planet. So I just asked my colleague Tom Fenchel, and we sat down together for a few days in my office, sketching literally with pencil and paper how these electron carriers work together, because they can't, as I said, they have to be paired. But it's more than just pairs. Now you have a network of reactions that are occurring across the planetary surface. And this is, if you go back to this paper in Science, this is to first order the reactions that lead to the generation or the consumption of the oxidized and reduced materials. So they're all related to each other with different metabolisms. About 90% of the metabolisms on this pokey little graph here or picture are anaerobic. That means there's no oxygen involved. And they look like this. So I'm just going to explain to you in a very simple way that if you were sitting in the London tubes at, for example, let's just take a very crowded station. So if we went to Piccadilly Circus, and you were on a crowded station, the train comes and you try to pack yourself in. Okay, so that train is now very reduced with respect to electrons. Think of yourself as an electron. You get now to the next station and electrons will leave the train. So the train is becoming more and more oxidized. And that's exactly how this works. This analogy is exactly how it works in nature. If something is already very reduced, it's very difficult to reduce it more. So that substrate is no longer made available to an organism that wants to reduce that substrate. Then some other guy comes along and oxidizes that substrate and makes it available for the guy that wanted to reduce it. Both guys are generating energy out of the system. Now, if I go back to this map, and I asked my former graduate student, Ben Yellen, to annotate it for me. How many genes are involved? There are only 400 genes responsible for all the electron transport reactions on the planet. Now let's think about this for a second. There are only 400 core genes. That's nuts. We've annotated millions of genes. So why are we annotating most of the stuff? It doesn't matter anymore. We know these 400 genes. Okay? So let's take a look, and I think about this from an engineering point of view, or a physicist point of view. Let's think about the entire surface of the planet as a circuit board. So we have one power supply, that's the sun. We have all these diodes and transistors on the planet that are converting the electrons from a substrate to a product, sometimes storing them, so they're a diode. And then you have wires. What are the wires? Well, there are two main wires on the planet. There's the atmosphere and the ocean. The oxygen you're breathing now wasn't generated in the room you're in. It wasn't generated in the city you're in today. It was generated somewhere else on the planet years ago. So it may have been generated in Brazil 20 years ago. So the atmosphere becomes a wire carrying substrates and products. The carbon dioxide you're breathing out is carried out into the atmosphere, and it is going somewhere else where it'll be consumed. The other wire is the ocean. So those are the two main wires. On very, 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 very long time scales, you could consider the upper mantle, which is also fluid, to be a wire. But to be honest with you, it's very difficult to see changes in redox conditions across long periods of geological time for markers that could explain how this wire works. For example, chromium or vanadium. Their redox states have been relatively constant for at least 3 billion years. So let's take a look at the metals that are moving the electrons. So it's not just iron or manganese, although there are quite a few substrates or enzymes that do that, but there's nickel, there's copper, and molybdenum. And to some extent, tungsten. Now, this has been explained in a few ways, most recently in a paper in PNAS, actually. Tungsten enzymes have been highly underappreciated. The mercury-containing enzymes are almost surely artifact. If anybody's interested in that, we can talk about it. But look, we don't have chromium-containing enzymes. We don't have, neodymium-containing enzymes. A lot of the potential redox reactions that could occur with other transition elements are not occurring because of their paucity. They're much rarer. So most of them are going to be generated by iron, manganese, a little bit by copper, and I'll show you that in a second. So this is the distribution of the metals in the oxidoreductases. And now, what is an oxidoreductase? We have 7 classes of enzymes, broadly speaking. And the class that moves the electrons, with or without the protons, or makes the hydrogen available through a hydrogen carrier, are called the oxidoreductases. It's EC1, the enzyme commission 1. So if I take they have 4 numbers. So enzyme commission 1.1.1, 1.1.1.1 is alcohol dehydrogenase. All of these have different numbers, but if it starts with an enzyme commission number 1, it's moving electrons or hydrogen. And so the guys that are moving the electrons, about 67, 68%, somewhere in this order, 69%, roughly 2/3, are using iron by itself with sulfur, for example, or making heme. And heme is iron within a matrix of an organic material, and heme is because it's red. And then you see copper and so on and so forth. Now let's just take a look at these guys for a second. This is the center of a very, very simple molecule. It's ferredoxin, which we all have, and it only does one thing. It moves electrons. That's all it does. Very simple. And now I want to get into something, because I'm talking to an Italian audience. I want you to understand, you're looking at a structure of the core of this protein, which only contains 28 amino acids in the half cell. And I want you to see 3 structures of the protein. So this is the protein backbone here. And you see that this is a twirl, and this is a flat type of structure, and then you have this squiggly type of structure. So I'm going to tell you, you can think of it in terms of pasta. If you're not familiar with the structures of protein, just think of it in terms of pasta. This is rotini. This is lasagna. And that's spaghetti. So you have these 3 structures you can throw into the pot to make these proteins: rotini, lasagna, or spaghetti. And we'll come to that in a bit. So here you can see the rotini. This is in another protein, nitrogenase. This is the enzyme, and there's only one type like this. And it takes nitrogen from the air and it makes ammonia. Without that, you wouldn't be here. So this enzyme is a very, very old enzyme, and you can see it's made up of many, many types of many iron sulfur clusters, 38 of them, in fact. And you can see the pastas are mostly helical here. You don't have much lasagna in this case. And you have this little bit of spaghetti. And here you have a lot of lasagna and no rotini. And this is another type of protein, and so on. We can go on like this. So we can arrange all of these oxidoreductases in a simple isosceles triangle. So this is spaghetti, 100% spaghetti, which no protein has. This is 100% lasagna, and this is 100% rotini. So you can just take all of those structures and figure out what percent of each of the pastas is in the core of the protein that's doing the work. All right? So why do we do that? Because now we can take from each of these proteins with that representation and draw Euclidean axes to every other one and determine distances. I don't care where these proteins came from. We can draw the distances from one to the other, to the other, to the other. And we're going to make a Bayesian hypothesis or a prior. So if you're not familiar with Bayesian analysis, don't worry about it for the moment. I'm just going to tell you an assumption. In Bayesian statistics, it's called a prior. And that is the most mutatable of the pastas is the spaghetti. It has the least amount of hydrogen bonds by far. And so the origin of these proteins had to be closer to this portion of this isosceles triangle than down here. And this is the distribution based upon the amount of hydrogen bond energies that stabilize the folds. So you can see exactly what I just said. Very, very few hydrogen bonds up here. This would be the primordial polypeptide. And then you have these much, much more rigid proteins that are having a lot of either the lasagna type material or the rotini type material. Now, why do I do that? Because then we can generate a phylogenetic tree of proteins based only on their structure. And you can see that that ferredoxin, I showed you, that very simple protein, 4 iron, 4 sulfur proteins, is very close to the root of the tree that we can see from extant proteins. And we can go on and on and on and on and on and on and on. And this is the guys that have a lot of pasta, and then we can get into the guys that have, a lot of spaghetti, guys that have a lot of lasagna, and the guys that have a lot of rotini. And here you come out to one of these proteins here, which contains copper, cytochrome C oxidase. And this is the protein in your body that is allowing you to reduce oxygen to water. So this is a very late evolved protein. It contains copper, 2 coppers actually. And you see we have very, very little copper up in the very beginning here. So copper is a late metal that was introduced much later on after the evolution of proteins that contained a lot of iron or nickel. Now let's take a look at those structures in a little bit more detail. And I'm just going to come up to, how am I doing for time? I have about 10 minutes, I guess. The processes of natural selection inhibit any change in a well-adapted system on which several other essential components depend. This is a paper by Margaret Dayhoff, and she was specifically focusing on ferredoxin. So once you make these things, they don't evolve very much. They're stuck. And there are only about 12 of those protein folds, these major motifs, in all the extant electron transfer reactions. So this is like a Lego set. You want to make something that moves electrons from water to oxygen, or you want to make something that moves electrons from nitrogen gas to ammonia. You go into the Lego set, you have 12 different Legos you can use. 12. You don't have hundreds. thousands. You have 12 Legos, and you can choose 6 of them, 7 of them. You can glue them together genetically, and now you can make very elaborate proteins. And these are the core proteins in the Lego box. Okay, so this one contains copper. This is the Lego set here. Put this little Legos over here, right? So here's the ferredoxin fold. This is a representation mathematically without the lines of the distances the structures are from each other. And this is the way we would reconstruct what the Legos were. We take a protein, extant protein, very complicated. It has iron sulfur clusters. It has all these other electron transfer reactions in it, these other minerals. So iron sulfur is obviously a mineral you know. It's pyrite, for example, greigite. And you can now take this and you can bust it up. And you can say, okay, I have molybdenum here, I have iron sulfur clusters, I have hemes, and I'm going to put them back together and rewire them. And how close are they to each other? And then you can make a certain number of modules, and you can turn those modules around and say, okay, what proteins have this kind of fold? Doesn't matter what this is. Rossmann fold. Here's the plastocyanin fold. I mean, the ferredoxin fold. So these are core folds. These are part of the 12. And I'm just going to tell you that there are 2 folds in the origins of life. The Rossmann fold, which doesn't matter to you what it is, and the ferredoxin fold. So we can go on like this. This is the Rossmann fold, the ferredoxin fold, and you can see them across genes. They're next to each other. And then you can see how the Rossmann folds became able to produce molecules that became hydrogen carriers. Here only carrying electrons. And then we can put those together and make artificial proteins. These never occurred in nature. Here's a 4 iron 4 sulfur cluster. Now it only has 12 amino acids. We made a 12 amino acid protein with an iron sulfur cluster core that is capable of moving electrons. This is a voltammetry experiment just showing moving electrons thousands and thousands of times. Now, what we've made here is I designed a protein from the motifs at the core of extant proteins. This protein doesn't exist in nature. We made it artificially. Why are we doing that? We're trying to engineer or reverse engineer what nature did to make an artificial hydrogenase, which we've done, to make an artificial nitrogenase, which we're doing, so we can make an artificial hydrogenase with 13 amino acids. We're trying to make an artificial nitrogenase with about 15 amino acids. And then I can put these little Legos together, and we can see how these can be put together, and you can see these motifs. Here's a lot of rotini, many rotinis, lots and lots and lots of rotinis. Okay? And this pathway here, this pathway is the first pathway that will fix carbon dioxide on the planet. It's called the Wood-Lungdal pathway. So we can see how these molecules from one side of the river, which we couldn't see before, can make more and more extensive molecules that get you to the other side of the river of metabolism. All right, I'm going to stop the lecture. I just want to say that we've discovered around 4,800 so far exoplanets. These are planets that are found outside of our solar system. They're usually around M stars, which are much, much weaker stars than our G-type star. So they're not around a sun-like star mostly, because we like to see these planetary bodies, and it's much easier when the glow of the star is much lower. That's number one. Number two, many of these planets are orbiting very close to their star. And the reason that we focus on that is because the closer they are to the star, the faster they go around the star, or inversely, the faster they go around the star, the closer they are to the star. And that means you have less telescope time to worry about. Telescope time is very, very, very competitive and very expensive. So we find a lot of these planets and planetary bodies that are too close to the star to be within the habitable zone. And many of them are gaseous bodies. So we know from the rate at which they go around the star, how close they are to the star, and from the wobble position of the Doppler shifts, the mass of the planetary body. So if we know the mass and the speed, we can calculate whether or not you can calculate the concentration of material within the planetary body. Is it gaseous or is it rocky? This is a very fascinating area of science at the moment. We're going to be launching several satellites in the next few years, and we'll discover many, 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 many more of these bodies. And here is the problem. Looking through the very, 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 very thin film of the atmosphere of that planetary body with the star in the background, we're trying to deduce the concentration, not what the gases are, the gas composition. So we have not yet found another planetary body outside of our solar system with significant amounts of oxygen. We're not really looking for oxygen, we're looking for ozone. So, but it's going to be very, very, very tedious. Sometime in your lifetime, and hope in my lifetime, we will find another planetary body with liquid water potentially on its surface, far enough away from the star that it's in the habitable zone, so it's not too hot, and potentially with gases that are far from thermodynamic equilibrium, like nitrous oxide and methane, or even ozone. And I'm going to stop there. Are there questions? How do I get questions?
Thank you very much, Paul, for the talk. I'll be relaying questions from the chats, from the chat on YouTube, or if anybody in the audience wants to ask a question. Let's see. Meanwhile, I have a question, Paul. I found your comment about how the mantle redox has not changed much over time. Well, you were speaking about the mantle being a possible wire for the Earth circuits. And we do reduced carbon in the mantle because of subduction. So we are bringing electrons down. Why is not getting any more reduced? Is because hydrogen is coming up from volcanic sources?
The amount of carbon, it's just the volume of rock in the mantle is super high. So even though you're bringing carbon down, it doesn't make really much of a detectable dent in the redox state of the mantle over even over 3.5 billion years. So I had this conversation when we were in Tokyo with Edward Schrock, but the person, if you really are interested in the data, there's a guy named Dante Canil, C-A-N-I-L, I think it is. Dante Canil is he is at University of Victoria, and he's been studying this for 30 years. So that's where you'll find the chromium and chromium valence states. I was surprised. I would have thought with all the oxygen on the surface of the planet, we would have found more oxidized chromium in the upper mantle. But no, it's it's extremely well buffered. It seems to be.
So it's both a buffer and a mass balance problem.
Exactly.
Mass problem.
Yeah.
Let's see. Any question for Paul?
I scared you?
Must be the time of the day, Paul. Let's see if I have anything.
Everybody wants to go have a little dinner and a glass of wine, I guess.
Well, I'm betting many of the people connected are actually having a glass of wine while you were speaking. At least I was.
Okay.
I can see David with his webcam on. Go ahead, David.
I'm wondering if you can get back to one of the first slides that you introduced about the concept of electricity in life.
Yeah.
Wait a minute. That one.
This one?
No, the next one. The one where you, at a certain point, you define. Here you go.
Vernadsky slide?
Yeah, and the one before. I've seen the slide, which is very very slow. The one before, where you showed the reaction between there is, here you go, oxygen synthetics.
Yeah.
Okay. Now, those are my understanding from, I'm not a great biochemist and other great biologist. I have just the fundamental in mind. But my question is that one.
Yeah.
What are the chances to see those two fundamental reaction working in other planets? What are the conditions to see this kind of situation in an exoplanet? I'm not saying evolved like the Earth system, but I mean, triggering those reactions in a way or another. How can we be able to trace if in a planet those two photosynthetic reactions are starting?
Before we got, this is an old question. So before we got oxygenic photosynthesis, there is still anoxygenic photosynthesis. So the substrates become hydrogen gas, which can be oxidized by photosynthetic organisms with light, H2S, hydrogen sulfide. And so now what you have is, if you're going to use hydrogen gas at high concentrations or hydrogen sulfide at high concentrations in the presence of light to oxidize the material, then you're almost certainly going to have to take your volcano with you every time you move around, right? If you have liquid water, you don't have to take your volcano with you. You can have water everywhere on this planet. So that's the great driving force for the evolution of oxygenic photosynthesis, which itself was derived from organisms that split H2S or H2 early on. So these are the purple, what's called non-sulfur bacteria, and the green sulfur bacteria. So those two organisms became the reaction centers of photosystems in the extant plants that we see. This type of machinery obviously was very complicated, and it still is. And the embarrassing part is we don't know when oxygenic photosynthesis first evolved. So, when cyanobacteria first evolved. So I would say we know that number within plus or minus 500 million years, and that's being a little bit generous, because I would also say we probably don't even know the number within about 800 million years. So then we have these proxies, right? We have proxies for oxygenic photosynthesis. And you would never find those proxies by telescope. So those proxies are going to be the oxidation of molybdenum, the oxidation of other trace elements. You'll never see that, by telescope. If you think about this, though, when I went way back here, when we first got this result, I emailed John Grotzinger, who was the chief scientist for the Mars missions at the time. And I said, John, that's why Mars is red. You've oxidized the siderite, the CO2 went off into the atmosphere, and you're left with a red regolith. And it had to have been only under aqueous phase. You could never do that. So we have these materials on the moon, our moon. Our moon isn't red. It's because it's dry.
Okay.
So this is an interesting phenomenon. How good the telescopes get to be. Can we see whether there's, at the end of the day, we're always limited by the number of photons a telescope can obtain. And so we're building these massive telescopes, we're trying to on Earth, 30-meter telescopes, because even though we have atmospheric problems and corrections, you can obtain far more photons from distant planets than you can with, say, the James Webb Telescope, which is not designed, was never designed for looking at extrasolar planets. So we're going to launch the sucker, but at the end of the day, it's going to be looking way into deep space for, very, very, very ancient galaxies.
Very clear. Thank you.
We have a couple of questions from YouTube. So first one is from Luca Tognetti. It's going back to nitrogenase, the different metals there. is asking if tungsten might have been substituting for molybdenum in the ancestral enzymes, given that molybdenum might not be abundant in the, presumably was not abundant in the Archean Ocean, despite having, you know, the phylogeny suggesting.
Yes. Hold on, I have to get my, I'm supposed to teach at 1 o'clock, so I'm just going to tell my class I'll be a bit late.
Yeah, we can still see your screen, Paul.
All right. So I'm sorry, Donato, what were you saying?
Luca was asking if you think that tungsten might have substituted for molybdenum in the Archean Ocean, given that molybdenum was likely not abundant, even if phylogeny suggests that molybdenum enzyme are actually oldest.
Yeah, of course. So molybdenum and tungsten can substitute for each other. And we've ignored tungsten mostly because we've looked at the most abundant enzymes. But there is a group at Georgia that is really interested in tungsten enzymes, and I'm surprised how many have been discovered in the last few years. So, I first became aware of tungsten as a substitute for molybdenum when Costantino Vetriani was telling me that there is I think it was a nitrogenase, not nitrogenase, it was a nitrate reductase.
I think it's a formate dehydrogenase, which is the same family of the nitrate reductase. Is
okay, so
one of those
tungsten, right? Yeah, I mean, the chemistry of the two metals is very similar in the D shell electron. So I suspect that, it would be very different. Obviously, we don't have a tungsten-based nitrogen fixing organism today that we know about.
Okay, yeah, survivor bias probably for some of the things. There's another interesting comment on YouTube by Gonzalo Ugarte Martinez. Apologies if I butchered the name. He's asking if the gene expression of the redox enzymes is also regulated by metals. So metals play a double role with the oxidoreductases. So
gene expression is a very, very complicated thing in prokaryotes. So we're only talking about prokaryotes here. And obviously, we have many ways in which we can regulate genes. If we're talking about transcriptional control, then clearly metals are involved, especially magnesium, which is not a true transition metal. Zinc, which is a true transition metal, but operates like calcium or magnesium. It doesn't operate as a redox factor. So you have zinc fingers and all these other metals there. At the end of the day, I've never explored that aspect of things, but yes, there could be a feedback or a do loop between metals and the expression of oxidoreductases, which I haven't looked at.
Okay. Then we have a question on the chat by Daniel Smurska, which is asking thank you for your talk. He says, if you can elaborate a bit on the point in which you say that even chemosynthesis is dependent on photosynthesis in the deep sea, considering origin of life idea about hydrothermal vents and the number of other reactions like methanogenesis that are not working on substrates that are derived from photosynthesis.
Yeah, okay. So we can go to the Mike Russell, Bill Martin world of hydrothermal vent origins. They like alkaline vents, which is fine. But what do they really like? They like the serpentinization reaction. That's what drives their philosophy, is the serpentinization reaction. So when I talk about present day hydrothermal vents, obviously the external world of a hydrothermal vent is oxidized compared to the reduced fluids that are coming out of the vent itself. And so the organisms like Riftia, which have those chemosynthetic bacteria that live inside, they're living on the redox gradient between the reduced materials and the oxidized materials. But the oxidized materials are available because the oxygen in the deep sea is gotten there by the subvection of water that had been to the surface and seen oxygen. So when you have a totally anoxic basin, and then you have hydrothermal vent fluids coming out, the redox gradient, there still could be a small redox gradient, but the operative word is small. So the amount of energy that you can capture from that system is extremely low.
So like Black Sea, for example, or
Exactly.
some of the...
Black Sea. Exactly. Exactly.
So basically limited by electron acceptors.
Exactly. Everybody has got an electron. Selling an electron is really hard. Buying an electron, nobody wants your stupid electrons. So that's why nitrogen gas becomes an electron acceptor, if you think about it.
Yeah, it becomes ammonia, absolutely. Let's
All
see
right,
if there's any. Yes, I don't see any other question for you, Paul, but they'll be in the chat. I'll let you know if there's any. Thank you very much for accepting again to give this talk. And look forward to you visiting us in Naples soon.
Okay, thank you. I hope so too. Bye-bye.
Bye, Paul. Bye-bye, everyone. Before you leave, I just want to tell you that we'll be meeting again on the 25th with another talk of the series. So I'll see you back here soon.